Thank you for having me here. And I'm excited to share our innovative technology um, and its application in bioimaging. So my name is Aranit Harizaj, and I lead the nanotechnology development at Trince. So today I will be introducing you to our technology that we have developed at Trince. The name of the technology is also Photoporation, and we will explore how it can be applied for bioimaging, and we will discuss its potential positive impact in the field of in the R&D field for bioimaging. Um, I'll just so I want to start by sharing a little bit of information about Trince. Trince stands for Transport into Cells. We are based in Belgium, in the city of Ghent, and we started as a spin-off company back in December 2021. So it's a Ghent University spin-off company, and so it's a little bit about 3.5 years ago that we started. And recently, we also won the prestigious Nature Spin-off Award prize, which really underscores our growth and advancement in the technology development, particularly in cell engineering and cell labeling. And at Trince, so we have developed a high throughput platform technology for non-viral transfection or delivery of different compounds in a variety of cells. And the technology, as I already said, is called photoporation. It is a combination of laser and nanotechnology to precisely make pores into the cell membrane. What really sets our technology apart from other state-of-the-art methods for delivery of compounds inside the cytosol of cells is the very high transfection or delivery yield. What do we mean with this? Is that we are the amount of positive cells for the cargo is very high, but also the viability is very, very high compared to other technologies. And beyond that, we can also do both adherent and suspension cells engineering. So you don't need to trypsinize your adherent cells to eventually perform the labeling step or delivering different compounds. You can do it on the adherent monolayer culture. Not only do the cells maintain a very high viability and high delivery yield, for instance, but more importantly, our technology is very gentle to the cells. The fitness or the cell fitness remains completely unaffected compared to the untreated cells. This means that you can move straight towards the next steps, whether that is, for instance, collecting the cells or performing different types of cell assays. Another advantage of our technology is the flexibility of recipients that you can use. So you can use different standard recipients, for instance, multiple well plate formats ranging from 6 well plate up to 384 well plates to eventually label your cells or transfect them. Standard plates that we typically use are 96 well plate, as this is the most convenient for typical cell experiments. Another key feature of our platform is that as we make use of a laser-based approach, we can label cells at a single or almost a single cell resolution. We have multiple devices ready. We have sent and shipped sold and shipped several devices. And our technology is also well documented with more than 40 publications on the photoporation technology and also around 8 patent applications filed. And since day one, we implemented the quality management system at Trince, which helps streamline the roadmap for its future CGMP device. So our technology has two main applications, mainly the R&D field for academia, biotech, or pharma, and the clinical use on, for instance, development of suspension cells, such as, for instance, CAR T cell development, or in adherent cells, such as the stem cells. We focus on modifying iPSCs via CRISPR-Cas or mesenchymal stem cells for clinical purposes. And the latter two, for instance, can be done without viral transduction using our technology, which eventually also reduces the cost and time for production. In the R&D applications, it's really would excel in screening applications, such as, for instance, screening CRISPR-Cas screening to find, for instance, the right guide RNA for the right target, as we can screen a lot of guides at once, or deliver various antibodies, nanobodies that are fluorescently labeled and eventually target subcellular structures for live cell imaging. It also allows for single cell transfection, and it enables to precisely deliver molecules to single cells for cell-cell communication experiments. This can be also interesting for spatial biology applications, where you just engineer a certain region of cells. So, as I mentioned earlier, our photoporation technology combines nanotechnology with laser technology to efficiently deliver various cargo molecules into the cytosol of cells. Now, let's take a closer look how it really precisely works. So first, we apply photothermal nanoparticles to the cells, which then attach to the cell membrane. So these nanoparticles will attach to the cell membrane. Then we apply a pulse laser, a nanosecond pulse laser, and we irradiate the whole well. These photothermal nanoparticles that are attached to the cell membrane will eventually absorb the laser light and convert it efficiently into heat energy. Because of the high energy input, the surrounding water molecules around the nanoparticles eventually evaporate, and there is a formation of a so-called vapor nanobubble. The vapor nanobubble is not stable and quickly collapses, releasing a mechanical energy. The mechanical energy uh, um, creates tiny pores in the cell membrane that close again very fast. Now, during that lifespan of these tiny transient pores, when your cargo molecule is freely diffusing in the extracellular medium, they can diffuse passively into the cytosol, and the pore closes again, and your cell is labeled or transfected as you need it. So now let's walk through a practical demonstration of how the photoporation process works in a lab setting. So first, the cells will be seeded in advance. If they are adherent cells, you seed them one day before, for instance, but you can also use suspension cells I already mentioned and continue with that process with suspension cells. Let's say we seed the cells in advance, we add a dilution of nanosensitizers to the cells, and eventually you have the interaction between particles with cells. After that, we add the payload to the cells, and the payload, what is important is that they are not connected to the nanosensitizer. So once you add the payload, you move on to the brief incubation, and you apply pulse laser radiation over the whole well. It takes around 4 seconds in a 96 well plate, 4 seconds per well to scan, and a whole 96 well plate is scanned in less than 10 minutes. So once you ready the whole well, then your molecules goes inside and the pores close again, and you finally have your labeled cells. And there is an optional washing or additions of the new fresh culture medium to the cells. So it practically goes very fast. So now let's take a closer look at some case studies, which I will be covering during this presentation. So I will be talking about photoporation and use in bioimaging. I will then I'll show some examples of spatial selective photoporation, so targeting almost single cells for cellular modifications. Then we will also have a look at technology that can be used to deliver gadolinium chelates, a contrast agent in MRI for in vivo cell tracking. And then finally, I also share some data on gentle transfection in stem cells, including examples of messenger RNA or Cas-RNPs. So let's start with the application of bioimaging. So here's an example of an experiment showing the delivery of fluorescent Dextran molecules inside the cytosol. And here's a confocal image which shows the cells labeled with the Dextran inside the cytosol. So we used a Dextran molecule of around 10 kilodalton labeled with a FITC to do an optimization experiment and as such to find an optimal concentration of nanosensitizers that we have to give to the cells and an optimal laser energy, because we can also fine-tune the laser energy. So you can see here some microscopy images where you see that by increasing the concentration of nanosensitizers, you get more positive or green cells, which is due to the fact that there are more pores created and more particles will interact with the cells, so more chance of creating pores. And here on the right side, you see an optimization experiment where we delivered the Physaction 10 in primary mesenchymal stem cells. And we did an optimization experiment where we used two different laser energies and different kind of concentrations or dilutions of nanoparticles. So from the left to the right, you have an increasing amount of particles that you add to the cells. What we can see is that we achieve almost a 90% delivery efficiency of these 10 kilodalton dextrins, which means that 90% of the cells are positive for labeling with dextrins. However, if you increase the concentration too far, then you see that the viability starts to drop, and it's a metabolic assay that we use. So you see, it shows you how many cells remain intact and properly eventually labeled with your dextrin molecules. And you see also some differences between two types of energies where you have for the same concentration a bit more delivery efficiency. So by fine-tuning the laser energy and nanosensitizer concentration, we can establish an optimal condition to ensure smooth progression for all the subsequent experiments. So you choose one concentration, one laser energy, and then you can do all the delivery experiments later on. So once the cells have been labeled with your fluorescent dextrins, or for instance, quantum dots or other labeling agents, you can move on for the next experiments. And here, for instance, this is a study where we did live cell imaging or in vivo imaging in mice. And in this study, we injected cells subcutaneously in the mice with control cells placed in the left leg and cells loaded either via incubation on the right or photoporation, as you can see on the right side. So always the control is on the left. And if you look at the images and quantify signal, you'll notice that the fluorescent signal of the control cells when you perform incubation of these fluorescent dextrins, so without photoporation, that the fluorescent signal is barely distinguishable. And even after quantification, you see that the signal remains very weak and very unclear difference between these two. While for photoporation, when you inject the photoporated cells, the fluorescent signal is significantly stronger and much more visible. And most importantly, cell tracking can even be effective up to 55 days. Well, you see some backgrounds or autofluorescence coming here, but you can clearly see the control cells not having any fluorescence, while the photoporated cells still have signal after 55 days, demonstrating the clear advantage of using our photoporation technology for cell labeling and performing longitudinal in vivo cell tracking. Another exciting application of our technology is delivering of nanobodies, so the nanobodies for intracellular labeling of various structures. For example, fluorescent nanobodies can specifically target mitochondria. They can target fission and histone structures. On the left side, you see where we delivered nanobodies against mitochondria targeting with the GFP label. And you can see them, so the cells are still alive, still moving, and you can do longitudinal microscopy imaging on live cells. The same for nanobodies targeting the fasciin, and eventually also histone, where we did a 24 hours follow-up of these cells. What makes this approach even more powerful is that the cells do not require any fixation, so they are alive. And this means that you can perform live cell microscopy imaging and track cells over an extended period of time, so enabling these long-term studies with minimal disruption of your structure of cells. As mentioned earlier, our laser technology allows as well to precisely scan the laser over the well. And this allows us to label cells in a highly controllable pattern, down to almost a single cell resolution. And with our current setup, users can in fact upload a specific structure or logo, which then guides the laser to scan and label cells in a predefined pattern with really great accuracy, as you can see on this picture, but I'll move on to the next slide. And in this study, we uploaded the image of the Einstein phase into our software, and the laser eventually will scan the dark region of this photo. And after scanning the well, for instance, where the cells plus particles are present, then you can see that only the cells where the laser has scanned will receive the dextran molecules directly into the cytosol. The black regions also contain cells. So this is a confocal image. It's a stitch of different images that we took. And these dark regions do not have the dextran inside, as the photoporation goes very quickly, and there is no time to take it even up. So there is a clear difference of signal. So we also can see that it's very detailed, and normally we also have videos where the cells still are moving and migrating. So you can see these videos in this publication from a while ago. And it just highlights that our technology is very precise, and it's a great approach for spatial selective delivery. In this study here on neurons, we used it to selectively target a neuron with phalloidin using photoporation. So we first labeled the surrounding cells with tubulin marker, and then we labeled one neuron with phalloidin, as you can see here in the merged or composite picture. So this demonstrates how photoporation really enables precise labeling even within a dense culture of neurons. We also did some multiplex delivery, where we first target in the first round of photoporation this cell here with a certain phalloidin with a certain Alexa Fluor 4088 nanometer label. And in the second photoporation step, we added another kind of marker, which is the Alexa Fluor 647, or the red phalloidin, as you can see here. So we can distinguish or deliver in 2 rounds 2 different kinds of markers or cargos. We also did this for two types of cells, where we delivered the phalloidin Alexa Fluor 488 in the neurons, and the astrocytes we then delivered another kind of phalloidin. So this highlights really the power of in achieving spatial selective labeling with photoporation. In this study, it's a little bit different compared to the fluorescent labels. Here we used our photoporation technology to label cells with gadobuterol. Gadobuterol is a clinically approved gadolinium chelate that is frequently used in patients. And what we did is we labeled the cells via pinocytosis, a form of endocytosis. And we also did it with photoporation, showing that we increased the amount of gadolinium atoms per cell detected by ICP-MS. And we checked if it's toxic to the cells. Are these gadolinium chelates, when present inside the cytosol or in the endosome for pinocytosis, is it toxic to the cells? And we saw that even for 100 millimolar, there is no toxicity observed. But what is more important is that when you just incubate the cells with gadobuterol, and you see that the concentration of gadolinium atoms per cell increases, there is no signal enhancement, as can be seen from this R1 quantified signal. And the same for when you look at the T1 weighted images, there is no signal enhancement, no widening of your image. However, when you deliver gadobuterol via photoporation inside the cytosol, you see a clear increase of white contrast on the T1 weighted image. And also when you quantify the signal, you see that it's increasing in function of the amount of gadolinium atoms per cell. So for the same amount of gadolinium atoms per cell, you don't have anything for pinocytic gadolinium, while for photophotoporated cells, you see a clear advantage. And this, in fact, for pinocytosis is something very well known, that you get a quenching effect, and that's why you don't see the signal enhancement. Next, we label SCOF3 IP1 cells using photoporation and injected them in different amount of quantity. So here it was 2 million, 4 million, and 8 million cells in the subperitoneal region. And the idea here was that we wanted to check if we can see signs of cancer invasion for these SCOF3 IP1 cells. We injected the cells, and you can see that the higher amount of cells, you get the higher amount of contrast, as you can see here. So it's around, bigger nodes. And what you saw is that you can track them until almost 16 days. So the cells, of course, divide and you will lose some signal. But here there was some sign of cancer invasion as depicted with red arrows in these regions, especially for the higher amount of cells, which is also logical. So this shows that we can even deliver different kind of contrast agents for different imaging modalities using our photoporation technology and enhancing um, the signal. We also injected, of course, the control cells on the right side, and there we never saw any signal for the control cells. So in this section, I will also be sharing some of our transfection results in primary stem cells, and these findings could also be of particular interest for the bioimaging community. And this will be on mRNA and COS-RNP transfections. So to start, we used our nanoparticles on bone marrow-derived mesenchymal stem cells, and we delivered messenger RNA encoding for EGFP expression. And what we saw is when we take several concentrations with one laser energy, we can see that we go up to 70% of transfection efficiency. However, the viability is still very high, so there is still room for improvement and increasing that efficiency even more. So this is up to the user to even go further and eventually find the maximum that they would like to have. We did also a Cas9 RNP transfection on murine MSCs. So these murine MSCs were continuously expressing GFP. And when we delivered Cas9, we saw that we can, with a certain concentration of nanoparticles, we can have a knockout efficiency of around 60%. But of course, your viability is still more than 80%. So there's still room for improvement and increasing that efficiency even further. What we can say is that we can, Photoporation can deliver COS-RNP efficiently into murine MSCs. And then lastly, also we can deliver COS-RNPs in primary human embryonic stem cells. And what we did is we compared our photoporation technology, which is here in blue-green, as depicted as Ben, and we compared the after photoporation how the proliferation kinetics is, and is it similar to the non-treated control in red? And indeed, we saw that the proliferation kinetics is the same as the non-treated control. However, when we performed electroporation with a commercial instrument, then we saw that for that optimized condition, the proliferation was hampered, and you had to wait several days until the cells started to grow again. So again, showing that functionality is preserved for our photoporation technology compared to the state-of-the-art technologies. What we also saw is that we can achieve around 60% of knockout for the interleukin-2 receptor on these primary human embryonic stem cells. Again, there is still room for improvement. You can go to 70, 80, or lower, depending on what the purpose of your experiments is. So basically, our photoporation technology can deliver a variety of cargo molecules, both into adherent cells as well as suspension cells. We have shown to be able to deliver these as well in tissues such as cornea and retina, and as well in amnion tissues. So most importantly for bioimaging, this can be quantum dots, polymers, fluorescent agents, gadolinium chelates, or other contrast agents for other imaging modalities. But nanobodies labeled with fluorescent probes can be as well being delivered for intracellular labeling. So to conclude, we at Trince have a benchtop system that works very efficiently, has a high transfection yield for different kind of cells and different kind of cargo. It is high throughput, so we can do a full 96 well plate in less than 10 minutes. We can do hard to transfect cells both in adherent as well as suspension cells, and delivery of various payloads such as RNPs, messenger RNA, siRNA, antibodies, nanobodies, gadolinium chelates, fluorophores can be delivered inside the cytosol of the cell. It's compatible with most of the standard well plates, even flasks, of T75 flask, for instance, and also slides. Micro dish can be also, depending what your substrate is, we can incorporate it inside the device as long as it's transparent to the laser light. There is no specific culture media required, so no specific transfection agent required for a photoporation technology, so you can use your own medium. And there is immediately a cell recovery post-photoporation. So thank you for attending my talk, and I'm open to receive questions from your side.